Imaging method reveals long-lived patterns in cells of the eye, a video by the National Eye Institute. Johnny Tam, PhD, head of the Clinical and Translational Imaging Unit at the National Eye Institute.
My lab is interested in imaging the eye, one of the only places where you can look inside the human body and see a complete tissue full of neurons, epithelial cells, and vasculature.
Eye animation.
When light enters the eye, it gets focused down to the back of the eye on a thin layer of tissue called the retina, where it gets detected by a specialized neuron called the photoreceptor. These photoreceptors don't exist all by themselves, but require the nourishment of a surrounding cell called the retinal pigment epithelial cell, or RPE. RPE cells historically have been very difficult to image because they contain pigment. And so any light that enters the eye gets captured and absorbed by the retinal pigment epithelial cells, making it difficult to image them using optical techniques. We've developed a new way to image the RPE based on the use of an existing FDA-approved dye called inoscinin green, or ICG. This dye is injected intravenously, where it travels up to the eye and is taken up rapidly by the RPE cells. After a relatively uniform uptake, it settles into a heterogeneous pattern with neighboring cells having different amounts of fluorescence. And this heterogeneity is what enables us to distinguish neighboring cells from each other.
Imaging data over 9 minutes.
Surprisingly, we found that with a repeat injection 1 year later in the same eye, that the same pattern was formed.
Data at 0 and 12
And
months.
so we could use this fluorescence signature as a way to track the eye across time at a cellular level.
Video of imaging setup.
To acquire these images, we use a technique called adaptive optics, which is a technique that allows us to image at cellular level resolution inside the eye.
Clinical center and patient.
Because we deploy our instrument in a clinical setting, we have access to a special population of patients, including a patient with Bietti crystalline dystrophy, a disease that is thought to affect the RPE.
Bietti crystalline dystrophy imaging data.
We found in a short time span of 7 months that there was a change in the fluorescence signature of the RPE, indicating that the RPE was likely to be disrupted in the early stages of disease. We're currently exploring different ways to translate this technology to the clinic where it can be more readily accessible.
Longitudinal adaptive optics fluorescence microscopy reveals cellular mosaicism in patients. By Zheng et al., published JCI Insight, March 21, 2019. National Eye Institute, nei.nih.gov, video produced by Leslie Earle.